A very good morning to all of you. So, as we'll continue, our topic is that of must-read topics. Previously, I have already uploaded 2 videos for paper 1 and paper 2. If you don't watch that video, so first go through that. What are the important topics for paper 1 and paper 2? So, let's discuss today's that are what are the must-read in paper 3. Paper 3 includes your molecular biology, your immunology, and your cancer, and your environmental biochemistry. Okay. So, what is important and must read for molecular biology? The first important thing in molecular biology for at the MD level is that whatever you will read, like replication, transcription, and translation, everything you will read at the eukaryotic level. Alright? Because prokaryotes is important for the UG, and for PG you must read at the eukaryotic level. First thing. Okay. So, first comes on the DNA replication. So, first important thing is that of DNA replication in eukaryotes. First question, LAQ. Okay. So, first is DNA replication in eukaryotes, and the question is also asked what is the difference between DNA replication in prokaryotes and eukaryotes. Okay. So, these two things you must read. After that, the important question is that of telomerase. Very, very important. The question is asked is what is the telomerase and telomere and what is its clinical significance. Okay, so you have to prepare according to that. And what is end replication problem? Or it can also ask in another way, what is end replication problem in your eukaryotes? Because end replication problem occur in the eukaryotes, not in prokaryotes. So, alright, so you have to prepare that. Next important LAQ is that of DNA repair mechanism. Very, very, very important topic, DNA repair mechanism. So, question is asked as a LAQ, what is DNA repair mechanism and what are the associated disorders of the DNA repair? Or individually, short note is asked for the xeroderma pigmentosa. Okay, so xeroderma pigmentosa you have to prepare as a short note also. Alright. Next important question is the short note, or it is attached with the LAQ of DNA replication. What are the inhibitors of your DNA replication and what is their clinical significance? Okay, so inhibitors of DNA replication either asked separately as a short note or it may be included in your LAQ. Okay. Next, the topic is that of DNA protein interaction. In DNA protein interaction, as such, DNA protein interaction is never ever asked before. The important in it, this topic is that of short note asked as the zinc finger motif and your leucine zipper. So, zinc finger motif and leucine zipper motif you read in this topic. Okay. Now comes on the transcription. So, transcription, as I told you before, transcription you have to read at the eukaryotes. Okay. So, what is the process of transcription? How it occurs in eukaryotes. Okay. And important thing is that of post-transcriptional modification. Okay, important. And what are the inhibitors of your transcription? An important topic is that of RNA editing and stability. Okay. So, these are the important topics in your transcription. Now, comes on the another topic, short note. Okay, it's important for your short note, that is of your genetic code and what are the characteristics of genetic codes. Prepare as a short note. Mutation and what is mutation and what are the different types of mutation, and other the short note, these are for the short note, that is what is mutation, what are the different types of mutation and what is difference between mutation and your gene polymorphism. Okay, so you should know the difference between mutation and gene polymorphism, and this difference is also frequently asked in your grand viva. So, you have basic idea what is difference between mutation and your gene polymorphism. Alright. Now comes on your translation. As translation, so you have to study what is the process of translation and how it occurs in the eukaryotes. Okay. Important thing is post-translational modification. Very, very important. Post-translational modification and post-transcriptional modification. Both you have to read. And individually ask short note on the post-transcriptional modification and individually short note ask on the post-translational modification. So, both these modifications are very, very important. Next important is that of protein shorting and protein targeting. And what are the disorders which are associated with your protein targeting? Next important question is protein folding. Very important topic, what is protein folding and what are the disorders associated with the misfolded protein. Okay. Alright. Next important question for short note is that of what are the inhibitors of translation in prokaryotes and eukaryotes and write their clinical significance. Remember, for inhibitors, whether inhibitors asked in for the replication, transcription or translation, you will write inhibitors in both. prokaryotic also and eukaryotic also. Okay, in that question you will write both for prokaryotes also as well as eukaryotes also. Okay. Next comes as a LAQ, very important, is that of the how gene expression or how the regulation of gene expression in prokaryotes and eukaryotes. So in prokaryotes you have to write your lac operon. A lac operon is asked simply as a short note also individually, and regulation of gene expression in eukaryotes is very important for a LAQ. Okay. And an important thing you have to read is that of CRISPR-Cas9. CRISPR-Cas9 is asked in it also, and CRISPR-Cas9 is very important for your fourth paper in recent advances also. So CRISPR-Cas9 is very important topic for both in molecular biology also, and in fourth paper in recent advances is also asked. So CRISPR-Cas9 is very important. And another important short note is that of chromatin remodeling. In lot of previous year question, chromatin remodeling is frequently asked. So must prepare topic is that of chromatin remodeling. Okay. Next is recombinant DNA technology. That is, what is recombinant DNA technology and writes its application in molecular medicine, LAQ. Okay. Now, important short note is that of Human Genome Project. Very important short note, Human Genome Project. And another short note important is that of gene therapy. Okay. So, Human Genome Project and gene therapy you must prepare as a short note and very important. Okay. Now, the next comes on the techniques. Okay. Techniques in molecular biology, techniques is either a part of your this third paper or it can also be asked in first paper also, because instrumentation and techniques is also in your under the syllabus of your paper 1. So you must go through it, prepare for the paper 1 as well as for paper 3. So the important molecular techniques you must read is that of PCR. First is PCR, conventional PCR, real-time PCR. Second is that of FISH, FISH. Fishes, that is fluorescence in situ hybridization. Important short note. Next important technique is that of DNA microarray. Okay, DNA microarray is also very important. Next is that of all blotting techniques. Southern blotting, northern blotting and western blotting. All three blotting techniques you must read as a techniques. Okay. Next is that of DNA sequencing. In DNA sequencing, the important DNA sequencing you read is first of that Sanger sequencing, important, and second is that of next generation sequencing. A lot of in previous year university exams, next generation sequencing asked many times. Okay, so next generation sequencing, and in next generation sequencing, because there are many types of next generation sequencing, you must read that is Illumina sequencing in next generation. So Illumina sequencing in next generation sequencing is a must read topic. Alright. Next important short note is that of DNA fingerprinting. Okay, how DNA fingerprinting is performed and what is its application of DNA fingerprinting. Okay, so these are the techniques, molecular techniques. So as I told you before, while you are preparing your exam and you go through your exam, so you must read it in your for paper 1 also as well as your paper 3 also. Okay. Next, bioinformatics. Very important question. Short note asked on the bioinformatics. Question can be asked, what is bioinformatics? What are the different tools of bioinformatics? What is the application of bioinformatics? Or short note, individually short note asked on the BLAST. BLAST. What is BLAST in bioinformatics? Okay, so what BLAST is one of the very important topic in bioinformatics. Okay, so BLAST. I will cover this bioinformatics in my upcoming video. So if you have any doubt, you can watch that videos. Okay, BLAST and bioinformatics, both I will cover that. Okay, next comes on the human genome. One important short note asked is that of satellite DNA. Okay, satellite DNA is also important topic. Okay, next important is mitochondrial disorder, also known as Oxford's disease. So mitochondrial disorder or Oxford's disease can be asked in paper 2 as a part of your electron transport chain, and it can also be asked in your paper 3. Okay, and I shared the video on Oxford's disease on my channel, so you can go through that. Okay, and important is that of nucleotide repeat expansion disorder. Nucleotide repeat disorders asked as a short note. Okay, And I will also share one of the video on it and trick also how you will remember all that. Okay. So nucleotide repeat expansion disorder, one of the important topic. Another topic, important topic for short note is that of imprinting disorder. Either question can be asked straightforward as what is the imprinting disorder, or short note can be asked in the Prader-Willi and Angelman syndrome. Okay. Because Prader-Willi and Angelman syndrome are example of your imprinting disorder. So either question can be asked directly as what is imprinting disorder or what is Prader-Willi and Angelman syndrome. Okay. Next is in one of exam the question is also asked what is the ethical and legal issue related to medical genetics. So just only read it. It's not very important, but just only take a view of that. That is what is ethical and legal issue related to medical genetics, because it is asked in one of the university exam. Okay. Now comes on the stem cell. Very, very important topic is stem cell. So you should know what is the stem cell, what is the clinical significance and medical importance of stem cell, and what are the types of stem cell. And in type of stem cell, you must know that is of iPSC. That is of induced pluripotent stem cell, as iPSC is asked separately as a short note. Okay. So in stem cell, you should know what is a stem cell, what is its clinical significance, what is application in medical field, and iPSC you prepare for as a short note. Okay. And also you have to prepare the ethical and legal issue related to use of stem cell in medicine. It is what is the ethical and legal issue related to your stem cells. Also you have to prepare. Now comes on the cancer. I already shared a series of cancer on my channel. That is important topic, so I already have covered. That is important topic. What is cancer characteristics? Never ever asked in MD. So important question is that of what is the molecular basis of cancer? Okay, proto-oncogene, tumor suppressor gene. Either LAQ will come, to LAQ question can be asked what is the molecular basis of cancer, or separately short note asked on the tumor suppressor gene, and separately question asked on the that was proto-oncogenes. Okay, and P53 is very important, and question can be asked why P53 is known as guardian of genome, explain. Okay, such kind of question can also be asked on the P53, or sometimes question can be asked why P53 is known as policeman of genome. That question can also be asked. Okay, so you know why P53 is known as guardian of genome or policeman of genome. Okay, next important is tumor marker. Very, very, very most important topic, tumor marker, as a short note. Okay. So you should know. Already I shared the trick how you remember the tumor marker. Okay, and next important short note is that of apoptosis. Okay, so apoptosis you have to cover in your paper 3, as well as apoptosis can be asked in your paper 1 also. Okay, so you while you will just going for the exam, so you must read it in paper 1 also and paper 3 also. Apoptosis, one of the very important topic. Okay, now comes on the immunology. Important topics in immunology is that of short note, write the structure, types and functions of different immunoglobulins. Okay, short note. So you have to write the structure, what are the types and what is the function of different immunoglobulins. Okay. Then important question is that of mechanism of antibody diversity. Very important. How the antibody are diverse in their mechanism. Okay. Next important is that of class switching. Very important topic, class switching. Okay, how the class switching occurs. Very important short note. And somatic hypermutation is asked frequently as a short note. What is somatic hypermutation and what is class switching? Okay, so you must read. And next is that of gene arrangement. How the immunoglobulin gene arrangement. So your like class switching is also asked in another type. What is isotype switching? So class switching and isotype switching both are same thing. Okay, so class switching and isotype switching is important, and your somatic hypermutation asked as a individually as a short note, and how the immunoglobulin gene arrangement is there. These can be asked separately as a short note, or LAQ can be asked what are the mechanism of antibody diversity. Alright, so you have to prepare that. Then humoral and cell mediated immunity. You should know what is the difference between humoral and cell mediated immunity. Okay. Next is that of MHC, very, very, very important topic in immunology, major histocompatibility complex. What is MHC? What is the role of MHC? What is the difference between MHC class 1 and MHC class 2? And how the antigen processing and presentation of MHC will be there. Okay, so these questions and these things you have to cover in your MHC, that is major histocompatibility complex. Then next is important is that of complement system. All the three complement system must read. Okay. Next comes on the hypersensitivity reaction, type 1, type 2, type 3, type 4. All type 4, 1, 2, 3, 4 must read topic hypersensitivity reaction. Then tolerance and your autoimmunity. Okay, tolerance and autoimmunity and your immunodeficiency. These are also important topic. And then vaccines, very important topic, vaccine, and you should know the DNA vaccines. Now, a DNA vaccine asked many times in question, previous year question, as a short note on DNA vaccine. Okay, so you have to prepare vaccine. First, vaccine comes as LAQ. So, what are the vaccines? What are different types of vaccine? How they are work? What are the advantages and disadvantages of vaccine? Or short note as a what is DNA vaccine? DNA vaccine asked frequently as a short note. Okay. Next question is the transplantation immunology. What is the role of immunology in transplantation? Okay. So it is not very transplantation immunology, although it's not very important, but in 1 or 2 previous year university exams, so you can just see it. What is transplantation immunology? That is not very at most important. Okay. So environmental biochemistry you can skip. Okay. In environmental biochemistry you can skip wherever ask the question in it. So this is all about the must read topics in your paper 3. So before going your university exam, these topics you must prepare. In another video, we will cover the important topics of your paper. Okay. I hope you just like the video. So thank you so much.